Hello, everyone, and welcome to today's broadcast, New PacBio Solutions for High-Resolution Microbiology and Immunology Analysis, presented by Jonas Korlach, Chief Scientific Officer at Pacific Biosciences. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by PacBio. For more information about our sponsor, visit them at www.pacb.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that Ask a Question box located on the far left of your screen and type the questions into the drop-down box that appear on the screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing this presentation, just click on the Ask a Question box and let us know you're having a problem. This presentation is educational and offers continuing education credits. Click on that Continuing Education Credits tab located on the top right corner of your presentation window and follow the process for obtaining those credits. Please join me now in welcoming our presenter, Dr. Jonas Korlach. I will now turn the presentation over to him. Welcome, Dr. Korlach.
Well, thank you very much, and thank you for LabRoots for allowing me to present again. It's been a few times now, and I've always enjoyed it. So thank you all for joining, and I'd like to give you today an update on new PacBio solutions for the areas of high-resolution microbiology and immunology analysis. I have the topics of the talk are listed here, so I'd like to start with a brief update on the PacBio sequencing performance as it builds the foundation for what I then spent most of my time on, which are the applications in microbiology in different areas, and I'll go into those in detail on genome sequencing, RNA sequencing, and epigenetics, a brief update on 16S metagenomics, and then also in terms of immunology, characterizing the immune repertoire and other associated applications. So with regard to an update on where we have come with PacBio sequencing, I want to start with this slide highlighting that since the beginning of the commercial phase of Pacific Biosciences, we have continuously increased the throughput of the system, of the sequencer, to the extent that now it's improved by over a factor of 1,000 compared to where we started. And so with the current throughput per SMRT cell run, you get up to 10 gigabases with the current chemistry for genomic DNA, and if you deal with amplicons, and I have some examples about that, up to 20 gigabases per SMRT cell. The instrument, the SQL system that you see in the middle there on the right bottom, allows you to run at present up to 12 SMRT cells. And so if you multiply that by 20 gigabases per SMRT cell, that's fairly serious throughput of about 200 gigabases or 0.2 terabases per machine run. And at the very end, I'll give an outlook showing that we're not done by any means and will increase the throughput even more. All of this is done at the foundation that we've always had of performance parameters that lend themselves for unique opportunities in the application areas, wherever sequencing, and I'll just today focus on microbiology and immunology. The average read length now is between 15 and 30 kilobases. The consensus accuracy, as you may be familiar with PacBio sequencing, is high because the errors are random. So we can achieve QV50 or 99.999% or less than one error in 100,000 bases consensus accuracy. PacBio has the least GC and sequence context bias, which means that it gives you very great uniformity of coverage and the ability to cover all of the sample, all of the genome, with the same kind of sequence quality. With PacBio, it is possible to directly infer epigenetic modifications like methylation and so forth. I mentioned that there's a flexible design, run design. You can run one SMRT cell or up to 12, and the acquisitions are flexible as well, depending on the application, between 30 minutes minimum and up to now 20 hours. So in terms of comparing that to other sequencing technologies, what we have tried to do, and hopefully I'll convince you that we've succeeded, is to build a system that is based on a foundation of quality. On the left in blue, I have listed the 4 performance characteristics that matter whenever you look at sequencing technologies. And what we wanted to build is a technology that has the best possible performance in all 4 areas simultaneously. As you know, other systems have made advances and are focusing on 1 or maybe 2 of those areas, and they have come at the expense of the other performance parameters. So the short read technologies have very high throughput and also high consensus accuracy, but they don't have the long reads, and they don't have the epigenetic characterization capabilities, and there's bias with regard to GC-rich DNA and so forth. And similarly, other long read or linked read technologies have deficiencies with regard to consensus accuracy, bias, some of them in terms of methylation detection. And so, of course, underlying this is the principle of being able to follow the polymerase that's now done on the Sequel system. So with that, I want to jump right in and highlight that as a result of these performance characteristics, there are now well over 4,000 peer-reviewed publications in all areas of science, and many of those are in the area of microbiology and also immunology. Currently, there's about 5 new papers on average per day that publish in the peer-reviewed literature that highlight the utilities and the benefits of single molecule real-time sequencing. And so what I'd like to do today is illustrate some published and some of them unpublished results on how this is being leveraged in the areas of microbiology and immunology. So starting with microbiology applications, one of the main use cases today is microbial genome assembly, de novo assembly. And a few years ago, there was a tweet at a conference that I liked, which said that I like how it's just assumed now that you use PacBio if you want to close a bacterial genome. And so I think by the community, PacBio is widely regarded as the new gold standard in bacterial genome assembly. And that's because of the performance characteristics that I mentioned. It is possible in a very robust and mature workflow now to get closed, complete bacterial genomes in one SMRT cell, and I'll later talk about multiplexing because the throughput is now so high. Not only getting the bacterial chromosome that you see on the left here, but significantly also resolving to completion and closing the plasmids, the mobile genetic elements that are in many cases so important in understanding the biology and understanding the pathogenicity of the bacteria. And so this has been made its way into the mainstream media. Here was an NBC News article from a while back where researchers at Walter Reed Institute over the weekend sequenced the complete genetic blueprint of one of these dangerous bacteria. And that has led to publications like these where it is now possible for the first time to really understand the evolution and generation of some of these very dangerous superbugs that are resistant to all known antibiotics. So this is a Klebsiella. And of course, where that plays a large role is in so-called hospital-associated infections. It's a huge problem in the US and worldwide because you have these superbugs that have this multi-drug resistance. This was a first paper quite a while now ago with Julie Segre and colleagues at the NIH Clinical Center looking at an outbreak there and highlighting that it is possible now with long-read sequencing from PacBio to get the full picture of these bacterial plasmids that allows you to track the transmission and follow the outbreak, which is not possible in the bottom with the short-read sequencing because the fragments are too short to give you that kind of visibility. And more recently, there was a follow-up paper looking at more of these types of bacterial strains in the hospital setting and finding, and this was again highlighted in the mainstream news, that there is a occurrence of these very dangerous bacteria, not only in patients, but also in the plumbing. In the sink drains are the bacteria that harbor some of these very dangerous multi-drug resistant plasmids, and then through horizontal gene transfer, that can then cause an outbreak. So this has already had some ramifications. The complete understanding using PacBio sequencing has had ramifications in hospitals rethinking and changing their hygiene procedures. There are many, and this is just a partial list of just searching our publication database. You see the link down there on the bottom, so you can look up all the papers that have published with PacBio. I've just searched for Klebsiella and hospital, I think, and this is just a partial list. There are many, many papers, too small to read, but I just wanted to give you a flavor that there are hundreds of papers now on this particular subject. I do want to give one little anecdote and case studies from a recent paper looking at Klebsiella pneumonia in hospitals with smart sequencing and showing how these PacBio closed genomes can reveal, both at the single nucleotide variant level and at the structure variation level, how they contribute to drug resistance and virulence. So this is a paper that was published in the Journal of Antimicrobial Chemotherapy by researchers in Germany, looking at the evolution of some of these very dangerous Klebsiella bacterial strains in a hospital setting using smart sequencing. They found that PacBio sequencing reveals both single nucleotide variation and structure variation. So these were different serotypes. They had multiple, as you see in the second bullet, multiple resistance to penicillins and other antibiotics. And they were able to find through the complete genome the different causative mechanisms, point mutations that make confer carbapenem susceptibility, resistance that is mediated by an indel, a 6-base insertion, and in one case, an 11,600 kilobase resistance gene that was lost from the other isolates that was conferring the gentamicin resistance. So here you see that with PacBio, it is possible to completely sequence and assemble these very complex plasmids that's enabled by smart sequencing. This was critical for characterizing the AMR, the antimicrobial resistance. And so the vast majority of antimicrobial resistance genes are always located on plasmids, and you can see the rearrangements and additional extra chromosomal elements that can come through bacteriophages and genes with high sequence similarity that change the properties of these bacteria. So here in one case, there is an insertion sequence repeat element mediated structure variant that's consolidating some of these antimicrobial resistance genes. So in summary, smart sequencing gives you unprecedented visibility into these processes that change these bacterial strains and change their level of danger and pathogenicity in the hospital setting. I want to just mention a couple other large-scale initiatives. This is the so-called Nctc 3000 project that was spearheaded by the Wellcome Sanger Institute in collaboration with Public Health England in the UK, sequencing and assembling with PacBio technology 3,000 strains, including some of the most dangerous bacteria. This found its way also in Business News Weekly and other mainstream media. Some of these are very iconic species. An isolate, Nctc1, was from World War I, from a soldier who died in World War I. The complete genome sequencing of the very original E. coli strains, some strains that were isolated by Alexander Fleming and so forth. And so this this project alone approximately doubled the number of complete finished bacterial genomes that were in the databases to date. Another example that I want to highlight is a paper that appeared a little while ago in Nature, looking at bacteria that were specifically killing male fly species. And of course, this has a significance in our attempts to control insect pests like mosquitoes, for example, Zika virus containing Aedes aegypti and so forth. And so in this paper, there was a long-standing sort of mystery of how these bacteria could just kill the male offspring, but not the females. And so this type of male killing capability had independently evolved in at least 6 bacterial taxa. And so there's one example that of this spiroplasma that kills all the male offspring in infected Drosophila flies. And this has been studied since the 1950s, but until this paper, the underlying mechanism had remained a mystery. Through the use of smart sequencing and the use of 2 strains that they found where one of the strains had reduced male-killing ability. The researchers were able to apply PacBio sequencing and compare the 2 strains to focus in on what might be the genomic region that is responsible for that capability. And so they recovered a plasmid that had a repeat-rich candidate gene that was then investigated further. And you can look at the paper. I won't go into this in detail, but this particular protein contained an ankyrin repeat that was shown then to be required for the host male-killing activity. And as a result, then, it was shown that it can now, this particular protein then targets these nuclear complexes that bind specifically to the male chromosome during development and then form, disrupting the DNA integrity and thereby killing the male offspring. And so really this thorough understanding of the functional ramifications was really only provided by PacBio sequencing and forms the foundation now for thinking about controlling insect populations. As I mentioned, the Gates Foundation, for example, has had first tests of Wolbachia-infected Aedes aegypti. Those are the mosquitoes. And that's another example for these male-killing symbionts to try to control the mosquito populations in Brazil as part of these virus outbreaks. So closing this section, just to mention that if you rely on the existing NCBI database entries to establish what you have in your lab, I want you to note that there's a certain risk associated with that because a number of the so-called complete genomes in the NCBI database that were done a few years ago contain, in some cases, quite dramatic errors. And so we've taken a survey a few years ago looking at about 20 or so strains that were all so-called reference strains, and resequenced them, sequenced them again, and then subjected them to a de novo assembly. And in this limited set, about 30% of those reference strain sequences had significant structural alterations that were either previous sequence errors or the bacterium has changed in the repository. So because it is now so straightforward and easy to redo that and do the genome from scratch, if you really want to be sure what you have in your tube or on your plate in terms of the bacterium, it's now quite easy to verify that and establish that using PacBio sequencing. So just an example of what it looks like. You can very easily see these kinds of discordances. On the top, you have the read structures if you have a concordant region, and then very clearly a deletion relative to the reference NCBI sequence where there was no coverage and the de novo assembly indicated a deletion. So to make this now more accessible and certainly to scale it concordantly with the sequel throughput, which is much higher than what you would need for one particular bacterial strain, we launched back in June our Microbial Multiplexing Kit that allows you to pool numerous bacteria in one sample and then sequence that in one reaction. So we have mature solutions all the way from the library preparation, smart sequencing, the data analysis, and the assembly. We have now verified barcoded adapter kits that allow you to multiplex up to 16 samples, optimized assembly parameters, and detailed recommendations on how you would have streamlined solutions for these types of multiplex de novo microbial genome assemblies. Currently, we recommend starting with about a total of 30 megabases of pan-genome sequence. So different bacteria have different genome sizes. So if you have something like H. pylori, which only has about 1.6 megabases of total genome size, you can consider, using all the 16 pooled species. If you have large bacterial genomes like Streptomyces has 9 or 10 megabase genomes, then maybe only you can pool 3 or so. So here are 2 use cases where different kind of genome sizes are pooled together. So we want to guide the users who want to use this to look at the approximate genome size and pool about 30 megabases worth of genomes. Also worth considering is the complexity of those genomes. Bacterial genomes have been classified, and the reference is on the bottom. So if you're interested in this in more detail, I would suggest you look at the paper by Sergei Koren and Adam Filippi, where the repeat content of bacterial genomes can vary drastically. If you have so-called class 1 genomes, they only have very few repeats. Class 3 genomes can have very large phage duplications and so forth. So depending on what type of bacteria you want to sequence, that can also have an influence on how many you want to pool. But then we have a microbial multiplexing calculator that makes it very easy to calculate to ensure equimolar pooling and even sequencing among the different isolates that you have in your pooled sample in the pooled SMRT cell, and detailed procedures and checklists for going through the process of the DNA library preparation, the sequencing, and the data analysis from the high-quality genomic DNA, shearing, and then the library SMRTbell preparation, all the way to the bioinformatic analysis in SMRT-Link. As I said, the initial experience we would recommend with 30 megabases, starting with pooled libraries. In some cases, it works well even with higher multiplex. So here in this example, 42.4 megabases of total microbial genomes was shown, used with the barcoded adapter kit and HGAP. And so in each case, you see on the second most right column, each of those genomes assembled into a single contig. And then the last column is the accuracy, the QV values. So all of them are at QV 50, 99.999% base accuracy or higher. So this is the workflow that we have in detail, which I mentioned is available in our documentation. And then we have detailed documentation and protocols on our website regarding this procedure. So now moving from the genome to the transcriptome, we've had a very pleasant collaboration with New England Biolabs, Lawrence Edwheeler, about adapting our IsoSeq, our full-length RNA-seq protocol, to bacterial RNA. Of course, they're not polyadenylated, so this particular preprint that is in the bioarchives and is now in press in Nature Communications, and I understand will publish this week sometime, details how we can adapt the protocol to bacterial transcripts. And so what you see on the bottom is just a screenshot example of a portion of E. coli. You can see the scale bar there on the bottom, 10 kilobases. So this is about 120 or so kilobases of the E. coli genome. And the skyscrapers that you see are the individual full-length transcript reads. So you see the operons here, and the paper describes how you have much better visibility now in the dynamic structure and regulation of operon transcription. So it's not the case, of course, that all the genes in a bacterial operon are transcribed in one piece. You can see very clearly on the bottom left here that the transcripts have different lengths. And in the paper, there's some examples that show that this is being regulated by the bacterium. All of this type of information is much more difficult to attain with short-read sequencing. And so it's really, for the first time, a detailed and unprecedented view on the complexity of the bacterial transcriptome, bacterial gene expression that was previously hidden before the ability of using this method. And then finally, the third pillar of understanding bacterial biology, of course, is the methylation, the epigenome. There are many, many papers now, well over 100 papers, that describe the unique ability of PacBio sequencing to fully characterize bacterial epigenomes. This has been reported and commented on in review papers, such as the one from Matt Walder a few years ago, entitled Entering the Era of Bacterial Epigenomics with Single Molecule Real-Time Sequencing. In particular, with regard to understanding the switching of certain bacterial pathogens between very deadly and very pathogenic versus not so dangerous bacteria, and how the methylomes and the methylation status sheds light onto these old enemies. On the bottom right, I've listed the entries. All of this information is now recorded in Rebase. That's the methylation database that's curated by Rich Roberts at New England Biolabs and colleagues. And you can see that there are many thousands of these entries from now over 1,200 organisms that have been subjected. Rich Roberts always asking me to encourage the scientific community, if you are sequencing bacteria with PacBio, even if you're not interested in the methylome, that's important biological information. So please submit that to the Rebase database. There's a very easy submission form, and so that the entire scientific community can take advantage of that information. It's been shown a few years ago, Matt Blow and others from the Joint Genome Institute did a very exhaustive and systematic survey showing that methylation is widespread among all classes and clades of bacteria, eubacteria, archaebacteria, and so forth. And the importance of methylation in bacteria has been highlighted in many papers. Here are a few that you can pause your recording and look up if you're interested, and also reviewed here in this latest review paper that concluded that smart DNA sequencing and methylome analysis has really revolutionized the field of bacterial epigenetics. So I wanted to highlight just a couple of examples that I thought were really nicely highlighting the utility and the value of getting at the methylation status of bacteria in this new and unique way. This was a paper 12 years ago from Japanese researchers looking at the effect of methylation on the lifestyle change of a bacterium, whether it's free living or then symbiontic in the nitrogen fixating nodules in legumes and other plants. And so, of course, back in 2006, there was no sequel system, and PacBio sequencing was not yet commercial, and so it wasn't possible to characterize the genomes. But what the researchers showed in this paper by bulk methods is that there are dramatic methylation changes as you go from the free living. You can see there's a dramatic increase in the signal intensity going to the nodule. And so I was always looking for researchers who would then go and apply this in conjunction with PacBio sequencing to get a higher resolution view of this phenomenon. And I was very pleased that a few years ago, Eric Triplett at the University of Florida did in fact this and looked at a particular soybean Rhizobium nitrogen fixating bacterium and exactly compared the different methylation status as the bacterium goes from a free living to a nitrogen fixing state. And they found drastic and widespread changes. 35.5% of all the genes were affected. There was a so-called symbiosis island, which you can see in the bottom figure, bracketed by the dotted lines, where there were over 10% of the methylation changes were in that island alone. And so there was some of the genes, 768 genes had significant expression changes. So the methylation then changes the gene expression. And so it really is fundamental to understand these dramatic lifestyle changes from these bacteria. The second example that I want to talk about is a paper, a very recent paper. It's a preprint now in the bioarchives, looking at the epigenetic fingerprinting with smart sequencing, enabling a new method and allowing to evade the natural defense systems of bacteria to allow for more efficient bacterial genetic engineering. And this is because, of course, bacteria have developed defense mechanisms, namely the restriction modification systems, that allow it to reject foreign DNA from invading that bacterium. So the chromosome of the bacterium is naturally methylated, and then the restriction enzymes will not digest the chromosome because the methylation protects, and any incoming DNA that does not have that methylation will be chopped up and neutralized. And of course, that's a big problem if you want to do molecular biology and transformations on these bacteria, because if your plasmid is not protected, then the bacteria will degrade it. And so the researchers here established a full pipeline to first understand the methylation, the methylome of the bacteria that you're looking at, and then creating transformation plasmids that can evade those natural defense systems. So they've developed this workflow where in step one, smart sequencing identifies the active restriction modification systems. That's then being incorporated into this genetic tool assembly and annotation. In step three, you can then, in the computer, design your plasmids and change the sequences to remove these recognition motifs. And then in step 4, de novo synthesize with gene synthesis these plasmids that are now having a much higher transformation efficiency, and that's shown here. In some cases, over 70,000-fold increase in the efficiency from the normal case, and so a much better transformation of this particular Staph aureus plasmid after the optimization of this procedure. So with that stealth by engineering approach, as the authors call it, facilitated and really enabled by smart sequencing, you can now expect to unlock quite a number of applications that pertain to bacterial genetic engineering, basic research, industrial microbiology, synthetic microbiology, and translational science. So I want to spend just a couple words about eukaryotic microbes. So, microbes in the largest sense of the definition is not just bacteria, but also microscopic eukaryotes. And so here's one example of Plasmodium falciparum, the malaria disease-causing agent. These are traditionally very difficult genomes to sequence because they're highly repetitive and extremely AT-rich. So conventional sequencing technologies are very challenged by this extreme skew of AT-rich sequencing. As I mentioned in the beginning of my talk, PacBio has very minimal bias with regard to the GC percentage, and so it's no problem to get high-quality data, to get even coverage. In this paper, the authors demonstrate that you can accurately and contiguously de novo assemble Plasmodium falciparum genome. The assembly yields all 14 chromosomes assembled end-to-end, and at the bottom figure, you see the read coverage is very uniform. So you get the complete genetic blueprint of this important pathogen. And then the second eukaryotic microbe that I want to highlight is a very recent paper in PLOS Pathogens that shows that a yeast strain that was previously considered both was both used in the food production and also was recognized as a pathogenic yeast is actually the same species. So there were 2 common but previously very poorly characterized yeast strains, and this overall reveals a potential public health issue because this Candida krusei is a commonly recognized culprit in clinical yeast infections. On the other hand, this Pichia species of yeast is classified by the US FDA as generally regarded as safe and is widely used in the production of biofuel compounds and also in food production. And so the authors wanted to understand the difference between those 2 yeast strains, and so they looked at the isolates of 20 of the clinical isolates and 12 of environmental isolates from the other yeast. And so this is described in this paper entitled Population Genomics, shows no distinction between pathogenic Candida and environmental Pichia, 1 species, 4 names. Because when the authors applied PacBio sequencing of those 2, quote-unquote, 2 yeast strains, they found, and again, it was very complete and accurate assemblies for both cases, which is shown in the paper. They found that the genomes were essentially identical. They were all collinear, and they were 99.6 identical at the base sequence. So according to that classification, they're basically containing the same species and are 2 different strains. And so if you don't have this particular comprehensive view on the entire genome and all the chromosomes and all the bases, it will be difficult to understand that how a Candida that causes disease in the clinic and basically the same species that's being used in food production is the same. So they looked at the isolates and their distance from each other. In red, you see the clinical isolates. In blue, you see the environmental strains, and then the susceptibility. And you see there's really no clustering. There are clades that have both members of the clinical strains and the environmental strains, so they do not form distinct clades. And so it is potentially very hazardous to use some of these isolates, especially when you think about immunocompromised workers and consumers. And so this is some really important insight that I think will have to be taken into account in future uses of organisms like this. So just a quick update. I know this focuses on microbiology, and back in April at the LabRoots meeting, I talked just about metagenomics, but that's a few months ago. So I did want to highlight one new paper, a preprint in the bioarchives about 16S sequencing. As a reminder, in PacBio sequencing, in addition to the standard sequencing that would be used in all the examples that I talked about until now, you sequence a bunch of molecules and then you build a multi-molecule consensus. Unique to PacBio sequencing is this way of doing single molecule consensus sequencing, or we call it circular consensus sequencing, or CCS, where by virtue of ligating these hairpin adapters to a linear double-stranded DNA molecule, it becomes topologically circular. And so when the polymerase starts sequencing, it will go around and around this molecule and sequence both the forward and the reverse strand of the same individual DNA molecule multiple times. It will generate what we call these subreads. One subread is one pass along one strand, and thereby you can get very highly accurate sequence from a single DNA molecule. That capability is not available with any other sequencing technology. It's unique to PacBio and has benefits in 16S sequencing, and then, as I'll also show you, in the immunology space and in many other spaces. Regardless of which type of sequencing mode you're using, you get the same kind of high consensus accuracy, and the curve is shown here. So at about a little over 20-fold coverage, you get to QV40 and so forth. So this is the preprint that I wanted to highlight by Benjamin Callahan and colleagues. We collaborated with them on refining the 16S ribosomal RNA gene sequencing with single nucleotide resolution and essentially no error. The paper concluded that the high resolution and accuracy they're reporting therein derives from the exceptional and maybe not entirely appreciated accuracy of PacBio CCS sequencing. That's why I want to highlight it here. And so what's remarkable, I think, in this paper is that each bacterium commonly harbors multiple copies of the 16S gene, and they tend to not be identical. And currently, with the Illumina short reads, you don't even look at the entire 16S gene, but this type of resolution is not attainable, and so you generally don't distinguish between the different copies. And because of the high resolution and accuracy and the single molecule nature of PacBio sequencing, for the first time that is now possible to identify and completely characterize each individual copy and count the copies in these different bacteria. And so this is shown on the right, on the lower right, where you see the bacillus on the upper left of that figure has 1, 2, 3, 4 copies that are there as singletons, and then 2 copies that have a slightly different sequence, and 4 copies that have yet another sequence. So 4, 2, that's 6, that's 10 copies altogether. And PacBio sequencing for the first time allows you to resolve all those in great detail. And it turns out that this previously had confounded the analysis and the databases. So also in the paper, which I encourage you to read if you're in this field, said that a lot of the PacBio data at first didn't look that accurate, but then upon further tests, it was actually the reference databases that are wrong, or smush all these different variants together, and now it actually shows that you can resolve that. There's another recent bioRxiv preprint that goes beyond the 16S application and shows that there are many applications for these high accuracy CCS long sequence read. That's Ben Merle at San Diego with new bioinformatic algorithms to denoise the long read amplicon data and get CCS at very high accuracy. So then now, in the last few minutes of my presentation today, I want to switch gears quite significantly and talk about immunology applications. And here, similarly, the CCS approach can be very powerful, of course, as an introduction to the immunoglobulin field and genes. It's of great interest to characterize antibody sequences. And of course, the diversity that's being generated comes through the VDG recombination with the random mixing the addition of non-templated bases. And then for B cells, you have the somatic hypermutation that can occur. And so that gives you this enormous diversity of antibodies that need to be characterized. And so what smart sequencing provides, which is shown here on the bottom, again, the long reads in conjunction with the highly accurate intramolecular consensus through the CCS capability allows you to get full-length reads on these very important regions. In some cases, they can exceed what you can span with the Illumina sequencer, especially for B cells, where you need the entire VDJ sequence to characterize the receptor and the repertoire. And so with smart sequencing, you can contiguously read across these entire variable regions. There's no imputation, no assembly required. You just get the direct full-length reads out. And now, whereas I would say previously, this was of limited utility because of the lower throughput that's not compatible with the high diversity. Now the SQL system brings a much higher throughput and the associated lower cost to really make that attractive and feasible. Historically, this application was spearheaded by Tim Smith from the USDA a long time ago now. In 2012, he had a paper showing that this can be done, and the principle on calves and cows that were either healthy or suffered from shipping disease. And you can already see just visually in the representation on the bottom right, you have different individuals with regard to their immune repertoire. And you can just visually see that there are dramatic differences between these individuals with regard to their diversity and how these cluster and so forth. And he also identified for the first time unusually long CDR3 sequences and so forth. Since then, there have been other examples for looking both in humans as well as in other animals, and including critically endangered primates, as is the Lemur paper, and then Steve Quake discovering new heavy chain antibody sequences using PacBio sequencing. And then one example that I want to highlight here is a paper by a group in the Netherlands about the B cell receptor repertoire sequencing. They've developed a method called artisan PCR, which has a much lower primer binding bias and gives a much better representation of the B cell repertoire. And so they use PacBio sequencing to identify and sequence directly full-length functional VDJ sequences in lymphoblastoid cell lines, the first bullet, in cryopreserved primary cells from various B cell malignancies and pre-malignancies, and then also in fresh frozen excisional biopsies that had B cell lymphomas. And the authors in this paper conclude that the massively parallel sequencing technology afforded by PacBio covers the entire VDJ sequences in a single read and provides an unbiased comprehensive analysis of these BC repertoires in healthy, reactive, and neoplastic conditions. They have since gone on and applied this technique to a number of different types of lymphomas, and the references are shown here. So if you're interested in this, I would encourage you to look up these articles in the journal Blood. This was a paper by Brendan Dukowski and other colleagues, Denny Duick at the NIH, looking at the linking of heavy and light chains. And because of the long reads from PacBio sequencing, you can directly amplify and sequence the linked heavy and light chains. And they identified extensive somatic hypermutations and rearrangements in these neutralizing antibodies that can now be phased across the heavy and light chains in single molecule reads. And then lastly, of course, we also know relatively little at this point about the underlying genomic locus, the IgH locus. And so just as a comparison to really what was a tour de force by Corey Watson in 2013, using back cloning and Sanger sequencing, quite a heroic effort to just elucidate and identify one more IgH locus in addition to the human genome reference. And so this was done in 2013. And just as a comparison, in the current whole genome human PacBio de novo assemblies, these regions fall out as fully contiguous sequences. So in the hydrodotiform moles, CHM1, CHM13, which are publicly available. The accessions are on the bottom. These IGH regions are fully contained in very large contigs. And then you can compare the CHM1 PacBio sequence to an Illumina and back-based approach that was also done on this very same sample. So this is an apples-to-apples comparison. In the Illumina work, this particular region was broken into 3 contigs on a scaffold. There were 2 large gaps, and there was a gap of 999 N bases, and then a larger gap that was arbitrarily set to 50,000 N bases. And so then with the PacBio assembly, it was possible to not only get the entire region in one contig, but also resolve the gaps. And you can see that the estimations of the gap sizes in many cases are off by a large degree. So this first Illumina 999 N gap was in fact 2,830 bases in size, and even more discrepant in terms of number of bases. The 50,000 base N gap was in fact occupied by 135,650 bases. So you get all the information with PacBio sequencing. Corey Watson, in collaboration with Melissa Smith at Mount Sinai, have now gone on and have published a poster and continue this work. The poster is fully available through this link on the bottom to identify and characterize the haplotype diversity of the immunoglobulin heavy chain locus across human populations, looking at ethnic diversity there, and looking at the full pan-genome diversity. And they use smart sequencing and assembly approaches for that. And so really highlighting how now this work brings this IGH research into the modern genomics era. And by doing this work and thereby further defining the full extent of the IGH diversity, they can now examine the impact that this has on antibody responses in disease. Just a couple of more notes, not only for humans, but also for animals, of course, and specifically in conservation biology. Resolving and characterizing the MHC gene family is of great importance. We were very pleased that this month's issue of the journal Genes highlights a paper that we collaborated on with Julian Sutton in Hawaii and others, Oliver Ryder from the San Diego Zoo, on a high-quality long-read de novo assembly of the last remaining crow species of the Hawaiian Islands. It's been extinct in the wild for many years and is undergoing an intense conservation effort of an in-captivity breeding program. And so with this genome, for the first time, it was possible to identify the full complement of all MHC genes, class 1 and class 2, and on the bottom, compare them to other crow species. And so this has implications on the breeding program to cross crows that have the highest amount of diversity, are the most fit with regard to disease susceptibility and so forth. And then finally, we were very honored to collaborate with researchers who maintain the IMGT database for single chain fragment variable, which of course are used in a phage display selection scheme for immunoglobulin or these SCFVs from combinatorial libraries for identifying new antibodies for novel targets. And the problem there and the challenge previously had been that the length of these particular DNA molecules is over 800 bases, but you really need the full length sequence for an absolute requirement for analyzing the associated heavy and light chain domains. And so this paper shows that there's a very large cost savings compared to what was previously done with Sanger sequencing, and a streamlined protocol in that the smart sequencing method in combination with this database and algorithm analysis provides now a much more straightforward protocol for characterizing these full-length SCFVs from these combinatorial phage libraries. So if you're in this area of research or want to learn more, I'd encourage you to look at this paper in Frontiers of Immunology. Also associated, of course, with immunology is the field of Hla typing. There are many papers on that. The first one on the left is a review paper, Hla typing for the next generation. I don't have time to talk about this because, just note full-length HLA, both class 1 and class 2, is very robust and mature now, is being used by companies like Histogenetics last year alone, running close to 500,000 samples on the PacBio system in their high volume HLA typing lab, and now even more throughput with the SQL system. And then, of course, also associated are the abilities to get a much better picture of viral populations, which of course are at the heart of the immune response. And so here are just a couple of examples of recent papers looking at these types of viral populations and subpopulations, quasi-species, in full length of high accuracy with PacBio sequencing. So with that, I'd like to close. I hope I've given you an overview and a breadth of the different applications and papers and approaches that people have used to give new meaning to some of these areas and provide new workflows and solutions. And I want to close, as I said, with the outlook, saying that we're certainly not done yet. I showed this slide before, and we continue to innovate and develop and increase the throughput. So this is an upcoming chemistry performance with the new targets. On the right, you see the current version 5.1, and we will again increase the throughput per SMRT cell, both for large insert libraries for genome assembly, as well as for amplicons. And as you can see, the new version 6 in the middle, for amplicons, there's a very quite dramatic increase in both the throughput per SMRT cell, up to 40 gigabases now, and up to 100 kilobases in average read lengths. And so that gives you even more power with this unique CCS approach. The table on the bottom shows that as a function of insert size on the leftmost column, you now get many, many more Q20 or Q30 reads, CCS reads, because the average read length is so much longer and the throughput is so much higher. So we're very excited about sharing these new updates. This is all in the context of our overall roadmap, where you can see here in the middle, scheduled for sometime in October, is this chemistry update that I was just talking about. Then late in 2018, very likely the version 2 Express library preparation. And then, as we've said many times, the introduction of a new SMRT cell, a chip, that will have an 8-fold increase, going from 1 million to 8 million features. And so we certainly project an additional order of magnitude throughput increase between today and over the next 12 months. And we're excited about the ever-increasing application spaces that this continuing performance and throughput increase will bring. So with that, I'd like to thank you very much for your attention, and I'd be happy to answer any questions. Thank you.
And thank you, Dr. Korlach, for that informative presentation and your important research. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box, drop-down box, and locate it on the far left of your presentation window, and type the question into the box that appears on that screen, and click that send button. Our speaker will follow up with those questions via email. So let's take a look at our viewers' questions. Our first question, Dr. Korlach, is for 16S, does resolution of the distinct 16S copies useful for identifying species, or does it just complicate analysis?
Yeah, certainly. So, you know, it is interesting. Over the years, I've heard, as CSO, I've heard a lot, oh, you know, this new information will make my analysis more complicated. And so now, of course, in many cases, you know, the researchers appreciate that it was precisely that new information that allowed them to make new discoveries and have new insights. So, yes, so in the 16S preprint that I highlighted, the authors note that their analysis was complicated by the fact that this was all new information and didn't match right away to the existing databases and reference sequences because they had lower resolution there. But my answer to that would be that it's just an amount of time. We've seen this over and over again with in bacterial sequences, with the complete plasmid resolution in the human space and in the immunology space, with new HLA types, with structural genetic variation and so forth. That's really critical to understand biology. So I anticipate that this new resolution, while maybe in the short term making the streamlined analysis a little bit more complicated, in the medium term will have enormous power, because now you have subspecies resolution. You can understand, you can even differentiate certain strains from one another. And I think that's very important if you think about not only hospital-associated pathogens, but also foodborne outbreaks. There are many species, Salmonella species, that are pervasive and commensal and not pathogenic. I understand something like 2/3 of all of the eggs that we buy in the supermarket have some form of Salmonella on them, but they're of course not outbreak causing and so forth. And so it's really this kind of resolution that will help the CDC and the FDA and researchers first, but then also the regulatory bodies, to really have more powerful tools to identify the pathogens early, to track their evolution, to understand resistance and so forth. So in the long term, I think it's going to be immensely powerful.
Thank you for that. And Dr. Korlach, this next question is related to syngenic DNA. There is differing transformation efficiency for plasmids grown in DCM plus versus DCM minus E. coli host. Why is that?
Oh yeah, you have very sharp eyes, and so I showed the example in the preprint of the Staph aureus plasmid, and so Staph aureus, so DCM plus of course is the 5-methylcytosine methylation, and that means DCM plus has it, and DCM minus is devoid of that. And so Staph aureus is one of the bacteria that have a so-called type 4 endonuclease, which specifically targets methylated. So remember in the introduction, the most common case is that the restriction enzymes are targeting unmethylated DNA, and methylation is used as protection. And then in this virus bacterium arms race, when there was a flip where now the endonuclease has the target is the methylated DNA. And so that is the reason why for these types of plasmids, you should use an E. coli that doesn't have that particular DCM methylation, so that it is protected against this particular type 4 Staph aureus endonuclease.
Thank you for that. And Dr. Korlach, would you like to provide our viewers with some closing remarks?
Yeah, certainly. So we would like to, I hope, as I said, I give you an update and an overview of all the different application spaces in the area of microbiology and immunology. I would like to encourage you to contact us, either me directly by email or through our website. There's local contacts in terms of field application specialists who can talk to you about your particular research questions, your applications, and to see whether smart sequencing can provide value for that. And so we look forward to these discussions, and I look forward to talking to you. Thank you.
And thank you, Dr. Jonas Korlach, for your presentation today. And I'd also like to thank Pacific Biosciences for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2018. You'll receive an email from LabRoots letting you know that the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now, and thanks for joining us. We hope to see you again soon. Bye-bye.